Welcome. In this session, I will be explaining about eukaryotic DNA replication termination stage. Okay. Termination of eukaryotic DNA replication includes telomere replication. Okay. At the end, telomeres are present, which will undergo telomere replication with the help of the enzyme telomerase, and termination occurs when the two replication forks collide with each other. So that time it will be halted, that is called stalled replication fork. With the help of topoisomerase 2, fork convergence also will occur during this replication process. So these are the events which will take place in the termination stage of eukaryotic replication. Okay, in the telomeres. So these telomeres are special structures that will seal both the ends of the chromosome. So you can find the telomeres or the ends of the chromosomes all sealed by a telomere cap called telomeres. Okay, this this is achieved with a by a telomeric repeat sequence, DNA repeat sequence. These telomeres are specialized structures which will have repeated G-rich sequences. So in the end, no, in the in the end of the DNA, you can find this telomere sequences T T A G G G T T. This is a telomere sequence. Okay, this telomere is composed of RNA molecule as well as enzyme telomerase. So this is both together. It will have this sequence as well as the uh, complementary sequence with the telomerase activity. So this TTAGGGGT means it will have this complementary sequence also. So for this corresponding complementary sequence also will be present in the telomerase. Okay, this telomere means it is composed of both the enzyme telomerase as well as with the telomere RNA sequence plus this enzyme telomerase is comprised together of this telomere. Okay, so this DNA sequences are this mammalian telomeric DNA sequences are randomly repeated base pairs, G-rich base pair TTAGGG. Okay, TTAGG means it's a mammalian sequence for telomeres. For vertebrates, you can find this sequence 5-TTAGGG for animals, birds, and reptiles, and all vertebrates. You can find telomeres having this G-rich repeated sequence TTAGGG. In yeast cell, you can find 5-TGT triple G. Okay, TGTGGG. In protozoan telomeres, you can find the sequence TTGGGG. So these are the telomeres which are identified in vertebrates, yeast cells, and in protozoans. Telomerase. Okay, so this telomerase is nothing but the specialized family. It belongs to a family of modified telomerase reverse transcriptase TERT, which is composed of a RNA molecule. So this here you can find at the end of the chromosome, you can find a telomere sequence. Okay, repeated sequence of telomeres. In this telomeres, this telomerase enzymes also will be encircled, which will have the complementary base pairing sequences. Okay, corresponding to the telomere sequence, this telomerase with RNA will have the complementary base sequencing. So during replication process, okay, when it comes to the end of the replication, DNA is synthesized in bidirectional mode. So one strand will be continuously synthesizing the leading strand. Okay, that strand is considered to be the leading strand, and the other opposite strand will be synthesizing Okazaki fragments, which is called the lagging strand. So in the lagging strand, involvement of many RNA primers are there. Okay, whereas in the leading strand, only one primer will be involved. So at the end of the replication, when this RNA primers are removed in the lagging strand and are replaced by DNA, the gaps should be closed by DNA ligase. Okay, however, 8 to 12 bases at the end of this lagging strand template cannot be synthesized because of the removal of this primer. Okay, due to the removal of this RNA primers, DNA polymerase cannot attach more nucleotides in the 5' end. Okay, it needs a free 3' OH end to attach the nucleotides. So the 8 to 12 bases will be lost at the chromosome ends. Okay, so at for each Okazaki fragments, this lagging strand will have many primers. So due to the removal of this RNA primers, 8 to 12 nucleotides will be lost at the chromosome ends. So shortening, okay, shortening of the DNA will occur in the lagging strand. That this shortening problem is said to be the N-terminal, that is end replication problem. Okay, so this eukaryotic cells will face a problem in the replicating ends due to the removal of RNA primers from the 5' end of the lagging strand, and RNA that DNA polymerase can add only nucleotides into the 3' OH end. So it will result in progressive shortening of both the daughter chromosomes. This problem is said to be end replication problem. So here, how this telomeres will help in this end shortening problem means this telomere regions and telomerase which are attached in the ends of these chromosomes will help to solve this end replication problem, because these telomeres can extend the uh, 3' end of the parental chromosomes. That means that it can add nucleotides from 3' to 5' end. That means it can add nucleotides at the 5' OH end. Okay, 5' end, not reverse direction. Okay, DNA polymerase can do can add that is base pairs, can add the nucleotide base pairs only from the 5' to 3' end, whereas the telomeres can elongate the chain length from the 3' to 5' end. Okay, so by in this way it will add this see here telomere is having the telomere is present TTAGGG. Okay, so the complementary base pair for this will be attached to this. So in this way, one by one the nucleotide will be elongated from 3' to 5' end. Okay, so the shortening chain length will be elongated with the help of this telomeres with the telomerase activity. This telomerase belong to the family of reverse transcriptase. Okay, telomerase reverse transcriptase TERT, which is having a RNA molecule with the complementary sequences corresponding to that of the telomeres. So in this way it will solve the problem of end replication problem. Okay, so this is how the chain end, that is the replication end terminal ends will be elongated with the help of this telomeres. Next is the termination. Okay, so eukaryotic replication termination occurs when the two replicating forks converge upon the same stretch of DNA. So when these two replication forks collide with each other, okay, it results in the halting or stalling. Okay, so DNA gets stalled or halted. When this collision occurs, no, it will get halted or stalled. So this topoisomerase 2 will resolve the topological stress to prevent the converging forks from stalling during termination. Under these conditions, stalling arises due to an inability to unwind the final stretch of DNA ahead of each fork. By promoting the fork convergence, topoisomerase 2 facilitate all downstream events of termination. So in the chromosome replication initiates at multiple replicons and terminates when the fork converge. 71 chromosomal termination regions TERs are identified and characterized. TERs contain fork passing element that influence fork progression and merging. Okay, this fork passing element is essential during collision. When collision occurs, the two replication, it will allow this in this region with the help of this RRM3 DNA helicase, okay, which will assist the fork progression across the TER region. So at this, when this replication forks collide with each other, RRM3 DNA helicase will assist any one of the fork to move through this passing element, and topoisomerase 2 associates at the TERs in the S phase and G2 facilitates fork fusion and prevents DNA breaks and genome rearrangements at the TER sites. In eukaryotes, replication termination appears to occur randomly within a 4 kilobase zone. So this replication fork frequently stall at centromeres, CEN, replication zones or SEZ, and tRNA genes and TY elements, and regions where collisions of transcription and replication occurs. So these are the regions where main collision of transcription and replication occurs. It occurs at the centromeres, CEN, and replication slow zones or SEZ, tRNA genes or TY elements. In this region only collision of the two replication forks will take place. The helicase RRM3, a component of the replisome, will facilitate fork progression through non-histone protein DNA complexes. Catenated intertwines can arise when two replicons fuse together. So it's a final stage of replication process where the two replication forks will converge and collide with each other upon the same stretch of DNA. So in the mammalian cells, even a single defective termination event would result in either DNA damage or mutation, or it will interfere with mitosis. This termination occurs when the two converging forks are apart 150 base pairs apart, at which it will create supercoils. That supercoils will no longer be resolved by topoisomerase. So when these supercoils cannot be resolved by this topoisomerases, termination occurs. Okay. So this will create a topological stress on the forks called pre-catenanes. So these catenanes, during this pre-catenane formation, fork convergence will take place. Okay, collision of the two replication forks will take place. Once the fork meet each other, the replisome will rapidly merge with each other over the replicated DNA. So this will allow the nascent strands of one of the fork to be ligated to the opposite fork. So at this point, the pre-catenanes will be called as catenated intertwines. Okay, these catenated intertwines arise when these two replicons fuse together. Okay, with the nascent strands in the opposing fork. So termination ultimately triggers to remove the replisomes by ATPase P97. That enzyme ATPase P97 is the enzyme which will remove the replisomes. So two replisomes are there. One will be in the leading strand and one will be in the lagging strand. So when both these replisomes collide with each other, when both the replication forks with the replisome collide with each other, it will result in the formation of pre-catenane, and when this one of the replication fork moves rapidly over the other, it will form in merging. That time that pre-catenanes will be called as catenated intertwines. Both topoisomerase 3 and topoisomerase 2 will help in the replication termination. Topoisomerase 3 has been involved in the resolution of this sister chromatid junctions which have been related to termination structure. Topoisomerase 2 will be associated with chromosomal regions during the S phase, and it is localized at CEN in the metaphase. Cell which lack topoisomerase 2 will experience DNA breakage upon cell division. So this is a model of replication termination. So in this you can find three stages: stage A, stage B and stage C. So in the first stage you can see the formation of pre-catenane resolution has been taking place. Okay, two replication forks with a replisome. So when they collide each other, it forms pre-catenane. Okay, topoisomerase 2 will mediate the fork progression at the TER zone by resolving the pre-catenanes behind the forks. Okay. See, RRM3 DNA helicase will helps in the progression of this replication fork to the pausing element. There are three elements. Pausing element, which is yellow. Okay, this pausing element is there. This pausing element is nothing but the polymerase alpha. Okay, then black color represents polymerase epsilon and polymerase delta. So one is responsible to synthesize leading strand and one is responsible to synthesize the lagging strand. So polymerase delta will be present in the leading strand and polymerase epsilon will be present in the lagging strand. So when they fuse together, okay, when they pass through these pausing elements, this topoisomerase 1 will help in the removal of the positive supercoils and topoisomerase 2 will help in the resolving the pre-catenanes, that is for convergence, okay, colliding of two forks. Second stage is fork fusion. So during the replication collision, polymerase delta will encounter polymerase epsilon in the opposing fork. So both the polymerases will collide each other. At least one of the two forks need to move across the pausing element. So for this, RRM3 will facilitate this fork progression at the pause site located within the TER. Then it will emerge with the asymmetric conformation, the leading polymerase and the lagging apparatus. Fork fusion will take place in the second stage. And third stage is catenation. Topoisomerase 2 then resolves the last catenation at TERs before DNA segregation, allowing the chromosome resolution. This is the third stage. Okay. So third stage is fork fusion will result in the formation of catenin formation. So this catenin formation will be removed with the help of the enzyme topoisomerase 2. Okay, so that it will allow the chromosome to separate during mitosis. So this is the event which takes place during the termination stage. So hope you would have understood about the mechanisms or what are the mechanisms which is involved in the termination mechanisms with that will control the replication termination and how this topoisomerase allows the replication with transcription and topological helps to remove the transcription difficulties of replication with transcription and how it solves the topological dynamics. Everything is clearly explained in this session. Thank you for watching this video. Stay tuned to watch more lectures in DSP Biochemistry channel. Subscribe and support the channel Biochemistry DSP. Thank you.